Okay, good noon to everyone. I would like to take this opportunity to introduce Dr. Shailendra in our midst. We welcome you, sir. In a defense of our student, Dr. Shailendra Kumar Arya presently holds a position of a professor at Department of Biotechnology, Engineering within the University Institute of Engineering Technology, Punjab University, Chandigarh. He attained his doctoral degree in biotechnology from the esteemed Indian Institute of Technology, Banaras Hindu University in 2010. With an affiliation dating back to 2006, Dr. Arya has been dedicated member of the Punjab University. His primary focus lies in the realm of the enzyme engineering, bioremediations, showcasing the profound expertise in enzyme wastewater treatment and sustainable development. Dr. Arya boasts an extensive publication record, contributing over the 80 research articles to the prestigious international journals. Furthermore, he has actively engaged in numerous international and the national conferences, where he has featured as a distinguished speaker, leading to a dynamic research group. Dr. Arya mentors both PhD and master students, guiding them in a diverse project encompassing enzyme engineering, wastewater treatment, algal study, and bioremediations. The collaborative efforts of his research team contribute significantly to advancing knowledge and the innovation in these critical areas. They have a lot of critical areas, discussion facilities, and online presentations, so many things. And he has biotechnology, PhD in biotechnology, intensive research, and he remained as associate professor. The professor presently is working in a Punjab University, and he has done he has done the 17 years of the research experience. So we welcome you once again, sir, and there's a presentation. Thank you so much. Can you stand up? Please.
Good morning, respected professionals. Good morning, my dear friends. Today I'm here presenting my final presentation of the thesis entitled CHALCONES Effect on Combating Antibiotic Resistance Due to NOR-A and AGR in Staphylococcus aureus. In this presentation, we will discuss introduction to the S. aureus and its antibiotic resistance, ciprofloxacin and mode of action, efflux pumps and its inhibition mechanisms, research gap, study of chalcones, ciprofloxacin combination versus Staphylococcus aureus, inhibitory effect of chalcones and ciprofloxacin on biofilm formation, and research outcomes. Starting with introduction part, as we all know, Staphylococcus aureus is a gram-positive bacterium that poses a significant threat in clinical setting due to its role in causing various infections. Staphylococcus aureus is a major cause of nosocomial infections and has become increasingly frightening due to its infectious potential. It causes life-threatening disease such as skin and soft tissue infections, bone infections like osteomyelitis and septic arthritis, and heart infections like endocarditis. The ability of S. aureus to form biofilms and develop multi-drug resistance is a critical factor in its pathogenicity. If we talk about the bacterium's characteristics, it is a gram-positive cocci. Sorry, slide is not, please. No, it's not working, please. ये इधर नहीं आ रहा.
Use this.
इधर ये ऊपर नीचे नहीं होता.
You need to use the mouse cursor. Hold on. Hold on, leave it.
Sorry.
That's okay.
ये Thank you. Should I continue? Thank you, sir. The bacterium characteristics, if we talk about its characteristic, it's a gram-positive cocci. The size ranges from 1 to 2 micrometer, and the bacteria are arranged in clusters, often described as a grape-like bunches. It is aerobic bacteria. The appearance of S. aureus on mannitol agar and its beta hemolysis on another agar plate, which is important for its identification in the laboratory. Come to the antibiotic resistance mechanisms of Staphylococcus aureus. This is a diagrammatic representation of the bacterial cell, highlighting several key mechanisms of antibiotic resistance. In this diagram, A represent efflux pump, which are protein in the cell membrane that pump antibiotics out of the cell, reducing their intracellular concentration and effectiveness. B shows inactivating enzymes such as a beta lactamases that degrade antibiotic, rendering them ineffective. C represents the target modification, when where the bacteria alter their target site of the antibiotic, making it less effective or completely ineffective. D represents target amplification, where the bacteria produce more of the target, overcoming the inhibitory effect of the antibiotic. E is the E-film formation facilitated by quorum sensing, a process where bacteria communicate to form a protective biofilm that shields them from antibiotics. F indicates decreased uptake, where the bacteria reduce the entry of antibiotic into the cell. And the G again highlights the role of efflux pumps in removing drugs from inside the cell. Come to the antibiotic agent we used in our study, that is ciprofloxacin. Ciprofloxacin is widely used antibiotic agent. It belongs to the chlorophenol class of antibiotics and is crucial in treating various bacterial infections. If we talk about its chemical structure, the chemical structure of the ciprofloxacin is complex, indicates its potency. It is classified under the fluoroquinolone class, which is known for its broad spectrum antibacterial activity. Ciprofloxacin acts by inhibiting bacterial DNA gyrase and topoisomerase 4 enzymes critical for DNA replication. This mechanism is bactericidal, meaning it kills bacteria rather than just inhibiting their growth. It is particularly effective against gram-negative bacteria, including strains like E. coli, Salmonella, Shigella, and Neisseria. If we talk about its therapeutic uses, ciprofloxacin is used to treat a variety of bacterial infections, including urinary tract infections, pneumonia, sexually transmitted infections, skin, bone, and joint infections, typhoid fever, gastrointestinal infections, lower respiratory tract infections, etc. Its broad spectrum of activity makes it is a versatile antibiotic. While ciprofloxacin is effective, it can be used. It can cause adverse effects. Common side effects are generally mild and include gastrointestinal disturbance like nausea and diarrhea. However, serious adverse effects can occur, such as a peripheral neuropathy, prolonged QT, interval seizures, and other CNS effects, hyper and hypoglycemia, photosensitivity, and tendinitis. These needs to be carefully managed. Let's talk about the efflux pumps. Efflux pumps are protein that play a vital role in removing toxic substances from cells. These pumps are developed in both prokaryotic and eukaryotic cells to protect them from harmful amphiphilic molecules. They act like a hydrophobic vacuum cleaners and ensuring cell survival in hostile environments. The diagram shows the structure and the function of efflux pumps, highlighting the various subfamilies. We have the major superfamilies major facilitator superfamily, small multi-drug resistance subfamily, multi-antimicrobial extrusion family, resistance modulation division superfamily, and ATP binding cassette transporters. Each of these subfamilies has a distinct mechanism of action, but they all contribute to removal of drug and toxins from cells. One notable example is a Norway family of efflux pump, which is found in Staphylococcus aureus. This well-studied efflux pump confers resistance to various antibiotics, including fluoroquinolones. The Norway pump is a significant concern in the context of antimicrobial resistance, and it can limit the effectiveness of these drugs. Efflux pump of Staphylococcus aureus, as we discussed earlier, it is a NOR-A, and it is a most studied pump in Staphylococcus aureus. It is encoded by the NOR-A gene, which produces a 42 kilodalton protein embedded in the bacterial cell membrane. This pump is chromosomally encoded and is expressed at a basal level under normal conditions. However, its overproduction can occur through two primary mechanisms, that is the mutation in the NOR-A encoding gene and the inducible expression of NOR-A via regulatory genes. If we talk about the substrate of NOR-A, it's an efflux pump, a wide range of compounds such as fluoroquinolones, biocides, dyes, quaternary ammonium compounds, and antiseptics. This broad spectrum substrate specificity make NOR-A a significant contributor to the multi-drug resistance phenotype observed in the Staphylococcus aureus. To counteract the effects of efflux pumps like NOR-A, researchers have identified several efflux pump inhibitors. The table on the slides lists some common efflux pump inhibitors, their sources and their mechanism of actions. First is Virampal. Virampal is a synthetic compound and is a calcium channel blocker, can inhibit the NOR-A efflux pump. Razapine, derived from Rauwolfia serpentina, act as a vesicular monoamine transporter and also can inhibit NOR-A. Other efflux pump inhibitors include carbonyl cyanide M-chlorophenyl hydrazone, a synthetic compound that inhibit oxidative phosphorylation, and piperine, a natural compound from Piper longum that act as the bioenhancers by inhibiting drug metabolizing enzymes. If we talk about the chalcones, they are alpha-beta unsaturated ketones, with benzyl-acetophenone being a significant member of their series. Chemically, they are characterized as 1,3-diphenyl-2-propene, 1-one, where two aromatic rings are connected by 3-carbon keto-ethylenic system. These compounds are not only synthesized but also occur naturally in various plant species such as Angelica, Glycyrrhiza, Humulus, and Secretariaria, which have been used in traditional folk remedies. Chalcones have been found to possess a wide range of biological activities, including anti-inflammatory, anti-parasitory, antidepressive, anticonvulsant, and antimicrobial and nitric oxide synthase inhibitory effects. The basic structure of chalcones is depicted here, showing the fundamental framework and a variant with hydroxyl group. The presence of hydroxyl groups can significantly influence their biological activity and solubility. If we talk about the literature, this is the literature review slides. These studies provide these studies provide evidence of the efficacy of chalcones in various applications, supporting the hypothesis that they could be valuable in developing new treatments and control measures. Motive of this our study is as our research proposal focus on understanding the complex mechanisms behind Staphylococcus aureus antibiotic resistance, particularly the interplay between efflux pumps and biofilm formation and the regulatory relationship between NOR-A and AGR system. Understanding these elements is crucial for developing effective countermeasures against this stubborn pathogens. In this study, we investigated the synergistic relationship between efflux pumps and biofilm formation in Staphylococcus aureus and their combined role in antibiotic resistance. Specifically, we explored the expression dynamic between the NOR-A efflux pump and the accessory gene regulator system. Our findings demonstrate that Staphylococcus aureus utilize the coordinated mechanisms to trigger resistance responses based on the external environment. By examining the interplay between NOR-A and AGR, we revealed a complex regulatory network that enhances antibiotic resistance. This research provides a critical insights into adaptive strategies employed by the Staphylococcus aureus to evade antibiotic therapy, shedding light on the potential targets of the development of novel antimicrobial therapies. These slides highlight the significant gaps that still exist in our understanding of the interaction between chalcone and the efflux pump in Staphylococcus aureus. The four key areas identified here are crucial for advancing our knowledge and developing effective therapeutic strategies. First, the exploration of chalcone on efflux pump inhibitors for NOR-A and other efflux pumps in Staphylococcus aureus is essential. Understanding how chalcones interact with efflux pumps, biofilms, and impact AGR gene regulation could provide a deeper insight into the mechanism of antibiotic resistance. Secondly, chalcones' effectiveness in enhancing antibiotic susceptibility is a promising area of research. Testing efficacy with different concentrations and in combination of various antibiotic is a logical next step for optimizing therapeutic applications. Thirdly, the correlation between the AGR gene regulation and NOR-A expression is an important area of investigating. Further mechanistic studies to understand this interplay are needed and understand the interplay between the virulence and the efflux pump activity. Lastly, the synergistic relationship between efflux pump and biofilm formation is critical for comprehending resistance development in Staphylococcus aureus. Understanding this relationship is vital for developing effective therapeutic strategies. Our study has three objectives. First is in vitro evaluation of chalcones in combination with ciprofloxacin against planktonic cells of Staphylococcus aureus. Second is in vitro evaluation of chalcone in combination with ciprofloxacin against biofilm associated Staphylococcus aureus. Third is the evaluate the effect of chalcones on AGR expression and NOR-A expression in sessile and biofilm forming Staphylococcus aureus using qRT-PCR. Moving to the first object, that is in vitro evaluation of chalcones in combination with ciprofloxacin against planktonic cells of Staphylococcus aureus. This slide shows chalcones screening and define their structure and molecular weights. As we examine the efficacy of chalcones in combination with ciprofloxacin against Staphylococcus aureus, ATCC1 199B, a strain that is known for nori overproduction, we observe a significant synergistic effect. The images present here depict the screening process where various chalcones were tested in conjunction with ciprofloxacin to determine their impact on the minimum inhibitory concentration of ciprofloxacin. And the table summarizes our finding showing the MIC of the ciprofloxacin when used alone and in combination with different chalcones. Notably, the combination of ciprofloxacin with C9 resulted in substantial reduction in MIC to 1 mg per liter, indicating a strong synergistic effect. This discovery is crucial and it highlights the potential of C9 as an adjuvant to enhance the efficacy of ciprofloxacin against bacterial infections. The images of the microtiter plates provide visual evidence of the bacterial growth inhibition, further supporting our results. Our research demonstrates that certain chalcones can significantly improve the effectiveness of ciprofloxacin, offering a promising avenue for addressing antibiotic resistance. Come to the MIC determination of the Staphylococcus aureus. This experiment utilized Muller-Hinton broth as the medium, following the broth checkerboard method. The setup between U-bottom 96 well microtiter plate sourced from Tarsan, India, with two-fold serial dilutions of ciprofloxacin ranging from 0.03 to 64 mg per liter and chalcone from 0.8 to 50 mg per liter. The inoculum was prepared using Staphylococcus aureus strain 1199B and 1199 with the density adjusted to 0.5 mcfarland and then diluted to 1:1000 in Muller-Hinton broth to achieve concentration of 5 into 10⁵ CFU per ml. The incubation was carried out at 37 degrees for 24 hours. The interpretation of the results was based on the minimal effective concentration of chalcone determined visually, and the lowest concentration of the antibiotic that showed no turbidity of the growth was considered as MIC. Moving to the results was based on the the in vitro studies on the combination of chalcone C9 and ciprofloxacin are presented in the table. The MIC of ciprofloxacin against Staphylococcus aureus strain SA1199, that is a wild strain, and SA1199B, that is nori overproducing strain, was evaluated with and without the test molecule. The findings indicates a significant reduction in the MIC of the ciprofloxacin when used in combination with chalcone C9, particularly at a concentration of 25 mg per liter, which is considered as the MEC. The results suggest that C9 potentiate the effect of ciprofloxacin, showing a maximum potentiation at 25 mg per liter. This is evident from the fold reduction in MIC values for the both S. aureus strains when ciprofloxacin was used in combination with C9. Next method used in objective 1 is agar diffusion method. The agar diffusion method technique is crucial for understanding the antimicrobial properties of chalcones derivative against Staphylococcus aureus ATCC strain 29213. The medium used was Muller-Hinton agar, and that is a standard choice for such assay. And we started with an overnight culture of Staphylococcus aureus ATCC 29213, standardizing to 0.5 mcfarland, which corresponds to a final bacterial inoculum of 10⁶ CFU per ml. This standardization ensures that our results are comparable across different experiments. Chalcone derivatives labeled as C1, C3, C4, C5, C7, C11, C13, C15 and C16 were suspended in DMSO at a concentration of 12.5, 25 and 50 mg per liter. These derivatives were then tested for their antimicrobial activity. For the agar plates, 200 microliter of adjusted bacterial culture was mixed with molten MHA poured into the Petri plates and allowed to set. Wells were punched using a cork border and 50 microliter of test concentration of chalcone derivative alone or 25 microliter of chalcone derivative combining with 25 microliter of ciprofloxacin, that is 5 mg per liter, were loaded into the wells. The plates were incubated overnight and at 37 degrees Celsius, the zone diameter of the inhibition was measured in millimeters to assess the antimicrobial activity. The results as shown in the agar diffusion assay indicates an increase in the zone of inhibition of ciprofloxacin in the presence of chalcone at different concentrations. Specifically, the zone of inhibition measurements were follows as C9 12.5 mg per liter showed no zone of inhibition, C9 at concentration of 25 mg per liter showed no zone of inhibition, and C9 at the concentration of 50 mg per liter showed no zone of inhibition. However, when combined with ciprofloxacin, the zone of inhibition increased to 16.5 mm for C9 at concentration of 12.5 mg per liter plus chalcone, and 18 mm for C9 at the concentration of 25 mg per liter plus chalcone, and 20 mm for C9 at 50 mg per liter plus chalcone as compared to 15.5 mm for ciprofloxacin alone. These findings suggest a synergistic effect between chalcone's derivative C9 and ciprofloxacin, enhancing the antimicrobial activity against Staphylococcus aureus. Next experiment used in objective 1 is chemical studies against Staphylococcus aureus. The experiment was designed to assess the bactericidal efficacy of the ciprofloxacin both alone and in the combination with chalcone C9 against Staphylococcus aureus. This studies utilize Mullihinton broth as the medium and an overnight culture grown suspended in sterile saline adjusted to 0.5 McFarland turbidity serve as the inoculum. The bacterial suspension was then inoculated onto the agar plates containing varying concentration of ciprofloxacin and chalcones. The plates were incubated at 37 degrees Celsius under shaking conditions. The CFU were determined by using serial dilution method at 2-hour intervals over an 8-hour period. The time kill curves demonstrated that bactericidal action of ciprofloxacin at a concentration of 2 mg per liter, which is 1/4 into MIC, when used in the combination with chalcone C9 at 25 mg per liter. And the graph shows a significant reduction in CFU per ml over time for the combination therapy compared to the ciprofloxacin alone. The data pointed on the graph represent the average log 10 CFU per ml plus SD of 3 readings, providing a clear indication of enhanced bactericidal effect achieving by combination with chalcone C9 against Staphylococcus aureus strain 1199B. This study highlights the potential of combination therapy in overcoming antibiotic resistance and achieving a more effective bactericidal action. Come to the next experiment, that is mutation prevention concentration of Staphylococcus aureus. This experiment or this step is a crucial in understanding how combat antibiotic resistance. The experiment was conducted using Mullahinton agar as the medium, where the inoculum was prepared with the bacterial solution containing 10 raised to power 10 CFU per ml of Staphylococcus aureus ATCC strain 29213. The 100 microliter of suspension was plated on MHA, ensuring controlled environment for the experiments. Agar plates were prepared with varying concentration of ciprofloxacin at different concentration of MIC with and without chalcone at the concentration of 12.5 mg per liter and 25 mg per liter. This step allowed us to assess the effect of chalcone on mutation frequency of ciprofloxacin against Staphylococcus aureus, and the plates were incubated at 37 degrees Celsius for 48 hours after spreading 100 microliter of the bacterial suspension on them, and the mutation frequency was calculated by dividing the number of colonies that appeared after 48 hours by the total CFU plated, and the results as shown in the table indicates the effect of chalcone C9 on the mutation frequency of ciprofloxacin against ATCC strain 29213. We observed that the combination of chalcone with ciprofloxacin significantly reduced the mutation frequency, especially at a higher concentration of ciprofloxacin. The optimal combination was found to be 25 mg per liter of chalcone C9 with greater than 4 into MIC of ciprofloxacin, which reduced the mutation frequency to less than 10 raised to power minus 9, thereby minimizing the emergence of resistance strain. And this study highlights the potential of chalcones in combination with ciprofloxacin to prevent the development of antibiotic resistance in Staphylococcus aureus. If we talk about the post-antibiotic effect, that is the effect of a drug against Staphylococcus aureus, that is the pharmacodynamic parameter that would be help in design of an optimal dosing schedule for an antimicrobial agent. The medium used was Mullahinton agar. The inoculum we used is a Staphylococcus aureus 1199B at 10 raised to power 6 FCFU per ml. Antibiotic exposure of ciprofloxacin at MIC concentration alone and in combination with chalcone C9 at 25 mg per liter for 2 hours, and the post-exposure treatment given to the samples were diluted to 1:1000 to remove the drug. In this incubation, the samples were taken every 30 minutes for CFU count until visible cloudiness was observed. For the calculation of post-antibiotic effect, we use the formula post-antibiotic effect is equal to T minus C, where the T is the time for the test culture to increase by log 10 CFU per ml after drug removal, and C is the time for the untreated control to increase by log 10 CFU per ml. And the table shows the different values of post-antibiotic effect, and this study revealed that in the conjunction with chalcone C9, ciprofloxacin was able to suppress the pathogen growth for longer period even after the removal of antibiotic, with longer suppression observed as the dose of antibiotic employed increased. Come to the second objective, that is in vitro evaluation of chalcones in combination with ciprofloxacin against biofilm forming Staphylococcus aureus. to calculate the inhibitory effect of chalcones and ciprofloxacin on biofilm formation, and the method used to assess this effect is based on the microdilution technique with the minor modifications. This approach is similar to the MIC assay used for the planktonic cells, where bacterial suspensions are prepared from overnight grown cultures and adjusted to a specific optimal density at 610 nanometers, corresponding to approximately 1 into 10 raised to power 9 CFU per ml. The experiment involves preparing 2 whole cereal dilutions of the chalcones in tryptone soya broth within 96 well flat bottom polystyrene tissue culture plate. Following this, fresh TSP and the bacterial suspensions are added to each well, resulting in a final inoculum of 6 into 10 raised to power 7 CFU per ml. The biofilm formation is then quantified by measuring the absorbance at 595 nanometer. This detailed methodology allows us to accurately assess the impact of chalcone and ciprofloxacin on biofilm formation, providing valuable insights into their potential applications. Come to the results of this study, that is chalcone in combination with ciprofloxacin against biofilm forming S. aureus, and the table presents the minimum biofilm inhibitory concentration of ciprofloxacin against Staphylococcus aureus ATCC strain 29213 when used alone and in combination with varying concentration of chalcone C9. As we can see, the MBIC of ciprofloxacin decreasing with increasing concentration of chalcone C9, specifically at 0 mg per liter of C9, the MBIC is 0.33 mg per liter, which reduced to 0.14 mg per liter when C9 is used at 50 mg per liter. This indicates a synergistic effect between ciprofloxacin and chalcone C9 against biofilm forming S. aureus. And the graph shows the synergistic effect by showing the absorbance values, which are indicative of biofilm formation at different concentrations of ciprofloxacin alone and in the combination with chalcone C9. The curves for the ciprofloxacin combined with C9 at various concentration that are 12.5 mg per liter, 25 mg per liter and 50 mg per liter shift to left compared to the ciprofloxacin alone, signifying a reduction in the ciprofloxacin concentration required to inhibit the biofilm formation when chalcone C9 is present. Notably, the combination with 50 microgram per liter of C9 shows most pronounced effect, indicating a potential therapeutic strategy to combat biofilm related infections. Moving to the effect of chalcone C9 and ciprofloxacin on Staphylococcus aureus strain assay 1199B biofilms. As observed through scanning electron microscopy, the SEM micro images revealed that a sub-inhibitory dose of 25 mg per liter chalcone C9 did not significantly impact a biofilm formation. Similarly, when the treatment with ciprofloxacin at 1/4 into MIC concentration resulted in a moderate reduction in the biofilm mass. However, the synergistic effect of combining C9 at 25 mg per liter and ciprofloxacin 1/4 into MIC was noteworthy, as it significantly inhibited Staphylococcus aureus SA1199B biofilm development compared to the untreated control. This suggests a potential therapeutic strategy for tackling biofilm related infections by combining to the untreated controls. These are the same results which provides a crucial visual evidence of the anti-biofilm effect on the Norway overproducing strain. The four images here represent different treatment conditions. Image A shows a treatment with the ciprofloxacin at 2 mg per liter, shows a reduction in biofilm cell mass. The combination of ciprofloxacin and chalcone C9 in image B results in a significant biofilm reduction. Inhibition chalcone C9 alone, as seen in image C, we do not have a noticeable effect, while the untreated control in image D exhibit dense biofilm formation. The key takeaway is the enhanced biofilm effect achieved by combining ciprofloxacin with chalcone C9, suggesting a promising approach for tackling biofilm related infections. Coming to the third objective, that is evaluate the effect of chalcone on AGR expression and in NORA expression in sessile and biofilm forming Staphylococcus aureus, is using QRT-PCR. Move on to the QRT-PCR methodology used in our study. We employed QRT-PCR to quantify the mRNA levels of the NORA and AGR genes in various strains, including ATCC strain, NORA overproducing strain, wild type strain, and a NORA knockout strain that are kindly provided by Dr. Nitin Palkalia from Naipur, Hyderabad. The transcriptional profile of NORA and AGR gene was analyzed in both free living cells and biofilm forming S. aureus cells. These cells were grown in a sub-inhibitory concentration of ciprofloxacin, specifically at 1/4 MIC, until they reach a mid-log phase. This was done both in the presence and absence of compound C9. For the QRT-PCR analysis, cells were harvested and RNA was extracted using triazole reagent. Following the manufacturer's instructions, the primer used were designed with Primer 3 software and commercially obtained. If we talk about the QRT-PCR results, they provide a critical insight into the expression level of the key genes associated with biofilm formation and antibiotic resistance in Staphylococcus aureus. The table explains the primer sequences and annealing temperatures, and where the list of primer sequences used for the gene 16S, NORA and AGR along with their respective annealing temperature. These primers were used to ensure specificity and optimal amplification during QRT-PCR process. The annealing temperature for all three genes is 62 degrees Celsius, indicating a standardized protocol for the experiment. If we talk about the graph, gene expression in planktonic versus biofilm cells, we observed the relative expression levels of AGR and NORA gene in planktonic and biofilm associated cells. The result shows a significant increase in the expression of both genes in biofilm associated cells compared to the planktonic cells. Specifically, AGR expression is increased by 2.97 fold, while NORA expression surged by 8.9 fold. This upregulation is consistent with the known roles of these genes in biofilm formation and the antibiotic resistance. The graph also shows the effect of chalcone C9 on the expression of these genes in biofilm associated cells. Upon treating with C9, the expression of both AGR-A and NORA was significantly downregulated, dropping to 0.05 and 0.42 respectively relative to planktonic cells. This indicates that C9 potentially inhibits the pathway associated with biofilm in formation and antibiotic resistance. And our results demonstrate the upregulation of AGR and NORE in biofilm formation associated Staphylococcus aureus and the ability of C9 to downregulate these genes, thereby inhibiting biofilm formation and antibiotic resistance. This study contributes to our understanding of molecular mechanism underlying biofilm resistance and open new avenues for the developing of novel therapeutic approaches. The molecular docking analysis of chalcone C9 compared with capsaicin of NORE and AGR inhibition. We utilize AutoDock Vina or molecular docking software to study the interaction of modeled NORE protein with capsaicin and C9. The result shows that the binding site residues at NORE are phenylalanine, leucine, methionine and isoleucine, phenylalanine 303 and arginine 3110 and phenylalanine 341. Notably, capsaicin and C9 exhibiting binding affinities of minus 6.77 kilocalorie per mole and minus 74 kilocalorie per mole respectively on the NORE protein. Furthermore, we investigate the effect of both ligands on the AGR gene cascade for the biofilm formation in Staphylococcus aureus using the PDB4BXI downloaded from the RCSB and prepared for docking study. This analysis provides insight into potential of C9 as more effective inhibitor compared to capsaicin. And the molecular docking sites for the molecules under investigation, as shown in the table, we have analyzed the binding residues and binding affinities of two molecules, capsaicin and C9, against two targets, NORE and AGR. And these results indicates that both molecules have significant interaction with target protein, suggesting potential therapeutic applications. And the difference in the binding affinities between capsaicin and C9 against NORE and AGR highlights the specificity of the interactions and the potential for designing more effective inhibitors. The binding interaction of C9, A shows the ball and stick model of NORE in green color and C9 in blue color. B shows the ribbon structure of NORE, that is green in color, and ribbon structure of C9 shown in blue color. And second picture shows the binding interaction of chalcones with AGR. C is the ball and stick model, AGR in yellow color and C9 in blue color. D is the ribbon structure of AGR with yellow color and chalcone in blue colors. And the binding affinities of capsaicin and C9 on AGR gene were 6.3 kilocalorie per mole and minus 6.3 kilocalorie per mole respectively. If we come to the summary, this study demonstrates the potential of chalcones derivative C9 as an efflux pump inhibitor in combination with ciprofloxacin against Staphylococcus aureus. And the checkerboard method revealed that C9 reduced the MIC of ciprofloxacin by 2 to 8-fold, with a notable 8-fold reduction among all the chalcone tested. The combination of ciprofloxacin and C9 exhibit enhanced bactericidal effects in kill kinetic studies and significantly reduce the emergence of resistance in mutation prevention concentration experiments. The combination of ciprofloxacin and C9 extended the post-antibiotic effect and inhibited biofilm formation at a minimum effective concentration as confirmed by the SEM analysis. NORA and AGR are involved in Staphylococcus aureus resistance due to efflux pump and biofilm development respectively. QRT-PCR result shows that the AGR and NORA expression were upregulated 2.9-fold and 8.9-fold respectively, and in biofilm associated cells compared to the planktonic cells. However, in the presence of C9, the expression of both genes was significantly downregulated. And the molecular docking study analysis reveals that C9 had a higher binding affinity to NORA compared to the capsaicin, that is a known efflux pump inhibitor. Come to the conclusion part. CHALCONES C9 reduce the MIC of ciprofloxacin by 2 to 8-fold against Staphylococcus aureus with notable 8-fold reduction, and shows enhanced bacterial sidal effect in the combination with ciprofloxacin, and the impact on resistance and biofilm formation, reduce emergence of resistance in mutation prevention concentration experiments, inhibited biofilm formation at effective concentration as confirmed by the SAM analysis. Effects of post-antibiotic effect, combination of ciprofloxacin and C9 extended post-antibiotic effect compared to the ciprofloxacin alone. If we talk about the gene expression and molecular docking, they shows the C9 downregulated AGR and NORA expression in biofilm associated cells, and C9 show higher binding affinity to NORA compared to the capsaicin, that is a known efflux pump inhibitor. Talk about the future perspectives. First is the in vivo studies to evaluate the efficacy and the safety of the C9 ciprofloxacin combination in animal models to validate its potential for the clinical applications. Broadening application to test the efficacy against the other antibiotic resistance Staphylococcus aureus strains or on other pathogens, and the therapeutic potential to assess potential of C9 or derivatives as an adjuvant to revive efficacy of existing antibiotic against resistant Staphylococcus aureus. These are the list of publications. We have 3 publications. First is in the review in. This is the review paper we have published. The research articles in African Journal of Biological Sciences. The second research articles in International Journal of Applied Pharmaceutics. The list of conferences attending during the study. The list of certificates. These are the reference used in our study. We have presented our acknowledgement to Dr. Sandeep Sharma for providing guidance, unwavering support and cooperation throughout the research, encouraged and motivated work on the research problem, instilling faith in carrying out the work, and Dr. Nitil Pal Kalia from the Department of Biological Sciences, Naipur, Hyderabad, which contributes expertise in pharmacology and toxicology and contributed strains of Staphylococcus aureus SA119B and 11999 NORA overexpressing, and to Dr. Gopal Karthik from the Department of Medicinal Chemistry from Naipur, Raebareli, who has synthesized and provide purely characterized chalcone derivatives. Thank you.
So now in our midst we have Dr. Adya. You can go ahead with the question. Any question?
Very nice presentation.
Thank you, sir.
So I have a few questions. Can you explain on the proposed molecular mechanism by which CHALCONES C9 down regulates both NORA and AGR gene expression?
So for this purpose, we use QRT-PCR studies to explain the upregulation and downregulation of the AGR gene and the NORA gene as compared to the planktonic cells and in the biofilm formation. That is QRT-PCR study.
Your QRT-PCR result shows significant down-regulated of NOR A and AGR genes. How do you confirm that this gene expression change translate into functional inhibition at the potential level?
If we talk about
Protein level.
If we talk about the we used a particular list of primers used in our study that helps us to identify their expressions on the protein level that are completely downregulated as we compare to the planktonic cells. That is to 0.42 and 0.53 microgram per liters.
Have you considered the pharmacokinetics and toxicity profile of CHALCONES C9?
So chalcones are, according to the previous research, they are chalcones are the promising compounds that possess relatively low toxicity and low stability. If we talk about in the toxicity aspects, they are relatively low.
In this thesis you have done.
So in vivo studies we will design.
Now question from second examiner. Why was ciprofloxacin chosen as the antibiotic for the combination therapy in this study?
So due to its broad spectrum activity that is used in we can use this as orally and we use this as an IV, and it possesses a broad spectrum activity to various compounds. That's why we choose this antibiotic.
Okay. What are the implication of your QRT-PCR finding regarding NOR A and AGR A expression in biofilm versus planktonic cells?
So our studies reveal that the upregulation of the biofilm formation as compared to the planktonic studies, which is 2.97 and 8.6 fold upregulated.
Okay. What would be the next step to bring the combination therapy closer to the clinical application?
So we can use these combination which we have studied used in clinical applications, and we will also try with another resistant strains of Staphylococcus aureus in clinical studies.
Okay. So question is over from both examiners. Now last question. What is your next plan?
Next plan.
We will definitely plan for the in vivo studies on animal models.
Over from my side. Any question from audience?
Students.
ये जो आपने स्टैफ ऑरियस यूज किया हुआ है, जिसको आपने इनहिबिशन दिखाई हुई है, ये स्टैंडर्ड स्ट्रेन है या पैथोजेनिक स्ट्रेन है?
सर ये ATCC strain है 2121.
ATCC strain है.
2921 strain.
Pathogenic or non-pathogenic?
So it's a non-pathogenic standardized strain.
तो एक आधा एक्सपेरिमेंट उससे भी लगा लेना बाद में कि जो पैथोजेनिक है, जो लोगों को तंग
So we will definitely doing in vivo studies.
ठीक है.
So I think everyone satisfied her presentation. According to my point of view, now you are eligible to awarded the PhD degree.
Thank you so much, sir. Thank you so much, sir.
Now we can say Dr. Bhawandeep Kaur.
Thank you so much, sir. Thank you, sir. Thank you. Thank you so much.
ठीक है जी, मुबारक है आपको।
Thank you so much.
करना